Hello everyone. I'm pleased to share with you this study published in iMeta, titled, Gut microbial-derived 3,4-dihydroxyphenylacetic acid ameliorates reproductive phenotype of polycystic ovary syndrome. Let me start with a brief background of the study. Polycystic ovary syndrome, PCOS, is one of the most common disorders in women. Current treatments are mostly symptomatic. Recent studies suggest that the gut microbiota and its metabolites play key roles in PCOS development. Interventions like probiotics or metabolite supplementation show promise in alleviating symptoms. Microbe-metabolite-host interactions may deepen our understanding of PCOS pathogenesis and offer new therapeutic targets. The highlight of this study. Multiomics bioinformatics analysis identifies bacterial and metabolite profiles associated with polycystic ovary syndrome. DHPAA alleviates polycystic ovary syndrome by inhibiting the BMP signaling pathway. Streptococcus thermophilus catabolites flavonoids-galactosidase to produce DHPAA. Gut microbiota play a crucial role in the effect of flavonoids against polycystic ovary syndrome. Next, I'll present our main findings. We recruited 30 PCOS patients and 28 healthy female volunteers as controls. Using untargeted metabolomic profiling and network analysis, we found distinct metabolic signatures between the PCOS and control group. Among the altered metabolites, DHPAA was significantly decreased in the PCOS group. Functional prediction analysis further suggested that DHPAA is closely linked to reproductive disorders. In subsequent animal experiments, we used two PCOS mouse models, Jiajia-induced and letrozole-induced, to evaluate the effects of DHPAA treatment. DHPAA significantly reduced the number of cystic follicles and serum testosterone levels, increased the number of corpora lutea in the ovaries, and restored the estrous cycle in treated mice, indicating its protective effect against PCOS. Ovarian transcriptome analysis revealed that the BMP signaling pathway, which plays a key role in follicle development, was significantly downregulated after administering DHPAA. This finding was further validated by mRNA expression analysis in ovarian tissues. We then treated PCOS model mice with a BMP pathway inhibitor and an agonist. Inhibition of the BMP pathway alleviated PCOS symptoms, while activation of the pathway blocked the beneficial effects of DHPAA. These results suggest that DHPAA may exert its therapeutic effects by suppressing the BMP signaling pathway. Through in vivo and in vitro bacterial culture experiments combined with targeted metabolomics, we confirmed that DHPAA is produced by gut microbial metabolism of flavonoids. Consistent with previous reports, we found that flavonoids could alleviate PCOS symptoms in mice. However, this protective effect was lost after gut microbiota depletion with antibiotic, indicating that flavonoid efficacy depends on the presence of gut microbes. In contrast, DHPAA retained its protective effect even after antibiotic treatment, suggesting that it may act as the final effector metabolite mediating the microbiota-dependent benefits of flavonoids in PCOS. To identify key microbes responsible for DHPAA production and PCOS symptom relief, we performed metagenomic sequencing and analysis on fecal samples from two independent human cohorts and animal models. Streptococcus thermophilus consistently emerged as a shared signal across datasets. In both in vitro cultures and in vivo bacterial treatment experiment, Streptococcus thermophilus was shown to produce DHPAA. Moreover, intervention with Streptococcus thermophilus significantly alleviated PCOS symptoms in mice. To further explore the link between Streptococcus thermophilus-derived DHPAA and its therapeutic effect on PCOS, we conducted genomic analysis of the bacterium and identified a gene encoding β-galactosidase, which is involved in flavonoid breakdown and DHPAA production. We then constructed a β-galactosidase knockout strain of Streptococcus thermophilus. In both in vivo and in vitro assays, the engineered strain failed to produce DHPAA. When administered to PCOS mice, this mutant strain showed no therapeutic effect. These results suggest that Streptococcus thermophilus may alleviate PCOS symptoms through β-galactosidase-mediated production of DHPAA. The conclusion of this study. DHPAA alleviates PCOS symptoms by inhibiting BMP signaling pathway. The protective effect of flavonoids against PCOS depends on the gut microbiota. Streptococcus thermophilus may alleviate the symptoms of PCOS by producing DHPAA via β-galactosidase. DHPAA and Streptococcus are candidates for the treatment of PCOS.